All right. Hello, everyone. Today we'll be presenting on skeletal muscle tissue engineering. So, skeletal tissue muscle engineering, or SMT, is an innovative field focusing on repairing or rebuilding muscle tissues that have been destroyed by injuries, surgeries, or diseases like muscular dystrophy. There are two main goals of SMTE. The first is to recreate muscle function. Scientists are currently working to create lab-grown muscle tissues that can actually integrate with a person's natural muscles, helping them regain movement and strength. To do this, they use materials that mimic real muscles and combine them with support cells that guide the repair and growth process. The second goal is to create muscle models for study. These are lab-based muscle tissues that help researchers better understand how muscles behave, both in healthy and disease. These models are incredibly useful for testing new treatments and studying the underlying mechanisms of muscle disorders. In short, SMT is both a therapeutic and research-driven approach to understanding and treating muscle damage. Let's take a closer look how skeletal muscles is organized, from the whole muscle down to the smallest functional unit. First, the whole muscle is made of a bunch of fascicles, which are small groups within the muscle. Each fascicle contains multiple muscle fibers, also known as a muscle cell. The muscle fibers are long, cylinder, and have multiple nuclei. They're packed with structures called myofibrils, which are essentially for muscle contraction. Inside the myofibrils are repeating units called sarcomeres. These are the smallest functional units of a muscle contraction and are what make muscle its striated or stripe appearance under a microscope. Sarcomeres are made up of two key proteins, actin and myosin, which slide past each other to produce movement. Surrounding these components of layers of connective tissue. Endomyosin surrounds each muscle fiber. Perimyosin wraps around each fascicle, and epimyosin encases the entire muscle. These connective tissues provide structure, protection, and support during muscle contraction. Altogether, these layered organization is what makes muscles to contract efficiently and withstand force. Next, we have this website. Let me share my screen. Confirm you guys can see it.
Yep.
So we are able to see how each muscle contracts all the way from a very big muscle to a very small actin myosin filament. I really like this website as it's very visual and helps me understand what is actually happening. Cross bridges are forming. It's sliding, releasing, forming, releasing, etc. And you can see on all different levels, and again, the muscle all the way down to the very small part. All right. To really understand how far skeletal muscle tissue engineering has come, it's helpful to look at its evolution over time. The timeline highlights key milestones that have shaped the field, from early foundation concepts to today's innovation. We begin with the 1950s to the 70s, where very first attempts were made to study and manipulate muscle tissue. At this point, the science were very early. Researchers were starting to understand how muscle tissues function, but actually engineering efforts were very limited in the understanding how muscle tissues function. Sorry. In the 1960s, the term tissue engineering first started to gain traction, though it mainly referred to general tissue repair and didn't focus on skeletal muscles specifically. Fast forward to the 1980s, a pivotal decade for muscle research, scientists made major strides in understanding myoblasts, which are the precursor cells that eventually become muscle fibers. They discovered that these myoblasts could be cultivated in vitro, meaning outside the body in a controlled lab environment. Even more exciting was the realization that these cells could be stimulated to differentiate or mature into functioning muscle fibers. In the 1990s, researchers advanced further with the development of a three-dimensional environment where muscle fibers could grow. This is where the use of scaffolds became a major focus. Scientists began to use biodegradable scaffolds that could mimic the extracellular matrix, which is the natural structure that supports cells in the body. These scaffolds allow for better organization of growing muscle tissues and could degrade over time, leaving behind newly formed tissue. In the 2000s brought even more innovation. During this decade, a range of biomaterials, including hydrogels and synthetic polymers, became available and were integrated into scaffold designs. These materials offer better biocompatibility and support for muscle cell attachment, growth, and differentiation. Research also began to explore mechanical and electrical stimulations during these culture process, essentially mimicking the natural environment of the working muscle in the body. These stimuli encourage the formation of more functional, contractile muscle tissues in the body. Finally, we reach the 2010s, the present day, where SMTE has become an incredibly dynamic and multidisciplinary field. The focus has expanded beyond just growing muscle fibers, developing more complex, functional muscle tissue constructs. Current research incorporates stem cell therapy, where stem cells are used to versatile cell source, as well as a gene editing technique like CRISPR to enhance tissue properties or correct genetic defects. Also, bioprinting, which allows researchers to print muscle tissues in highly organized structures. Today's efforts are centered on engineering fully functioning vascular muscle passage that can be implemented into the body to create a variety of conditions, such as muscle dystrophy, traumatic injuries, and muscle wasting disease. The goal is to create muscle tissues that not only look like the real thing, but behave like it too, contracting, integrating, and responding like a muscle. So why skeletal muscle tissue engineering matter? Musculoskeletal conditions are one of the key leading disabilities worldwide. In fact, over 1.7 billion people are currently affected, with an enormous implication not only for individuals' health, but for the economy. In the United States alone, these disorders resulted in 213 billion annual costs due to the treatment, loss of productivity, and long-term care. Some of the most serious muscle-related conditions include muscle dystrophy, myopathies, sarcopenia, and ALS. We do have a range of treatment options, including corticosteroid and gene therapy to physical therapy and orthopedic surgery. These approaches come with high costs, significant side effects, and limited long-term effectiveness. This brings us to the critical challenge, the need for more affordable, safe, and targeted therapy that can provide lasting benefits. There's also growing interest in personalized therapy that account for a patient's unique genetic and disease profile. With the global population aging and demand for better treatment increasing, the musculoskeletal therapy market is growing rapidly. This makes it now a vital time to invest in and explore innovative approaches like skeletal muscle tissue engineering, which could reshape how we treat these conditions. Now onto my research paper. My research paper is titled Tissue Engineering Applied to Skeletal Muscle, Strategies and Perspective. There are four major topics that we will cover. To engineering functional skeletal muscle tissues, it's essential to start with the cells that have potential to differentiate into muscles. There are four major types of cells that we can use for this purpose. Satellite cells, which are the native stem cells found in skeletal muscles. They can become activated in response to injury. Once activated, they can proliferate, differentiate, and fuse to form new muscle fibers. Because of this, they're considered the gold standard for muscle regeneration. However, their numbers are limited, and isolating them from patient is invasive and technically challenging. Second is embryonic stem cells. ESC are pluripotent cells derived from early-stage embryos. They can be differentiated into cell types in the body, including muscle cells. Their advantages lies in their high plasticity, but their use is limited by ethical concerns. Next, we have induced pluripotent stem cells, iPSC. iPSC are adult cells reprogrammed back to the pluripotent state. They may have many properties like ESC, including the ability to become muscle cells. iPSC are exciting because they can be patient-specific, reducing the risk of immune rejection. However, challenges include the need for better control over this differentiation. And then last, we have myocytal stem cells, MSC. MSCs are multipotent cells found in bone marrow, adipose tissue, and other sources. They're easy to isolate than satellite cells and can support muscle regeneration through differentiation and signaling. However, their ability to form mature myofibers is still under investigation and may depend on the microenvironment and the implied stimuli. In muscle tissue engineering, we use biomaterials to build scaffolds that can help cells grow and form new tissues. We can use natural polymers like collagen and gelatin that come from the body and are great for supporting cell growth, but they can break down quickly or be inconsistent. We can also do synthetic polymers like PLGA or PCL are man-made. We control their strength and shape, but they don't naturally support cells unless we modify them. Hydrogels are also available as a soft gel-like material that can hold a lot of water, perfect for mimicking muscles. They can also be mixtured with cells during printing or casting. And decellularized extracellular matrix come from real tissues with all the cells removed. They keep the natural structure and signaling that help the cell grow and organize properly, making them the most realistic scaffold option. We can also look for techniques for mimicking the muscle environment. We can use electrical spinning creates thin, aligned fibers like spaghetti that guide muscle cells to grow in straight lines, just like real muscles. We can use rotatory jet splitting is a fast version of the fiber production that makes a line structure. It's good for making large amounts of scaffold quickly. And 3D bioprinting lets us use precisely placed cells and materials layer by layer, even creating complex muscle shapes and fiber patterns. It's one of the most advanced and customized method we have today. Lastly, functional tissue engineering regeneration faces several challenges that impact long-term success. First, matching scaffold degradation with the rate of new tissue formation is critical. If the scaffold degrades too quickly, it may be compromised structure integrity. If it degrades too slowly, it can hinder tissue remodeling. Second, the immune response can trigger inflammation that impairs healing or leads to a scalpel rejection. Designing biomaterials that modulate or avoid adverse immune reactions remain a major hurdle. Third, insignificant vascularization or integrated often limits functional in engineering tissues, especially in the muscle, nerve, or complex organs that heavily rely on these networks. Lastly, ethical and regulatory issues arise with certain cell types, such as the embryonic stem cells or the genetically modified cells, posing barriers to clinical translation. Now onto my research paper titled Engineering 3D Skeletal Tissues with Complex Multipinnated Myofiber Architecture. Skeletal muscles isn't just a bunch of cells, it's a carefully organized structure. From the nanometer scale of a sarcomere to the macrostructure of a muscle fiber, each layer plays a role in contracting and force transmission. Different muscles in the body serve different mechanical needs. Some are structured for speed, others for force, depending on the architect. But when major injuries like voluntary muscle loss occur, the body can't rebuild these complex structures because the ECM and the progenitory cells are destroyed. Treatments like muscle grafts help, but often lead to fibrosis, reduced function, and even donor site damage. The challenge is to rebuild muscles that look, align, and behave like the original, and that is what the study is hoping to address. In the study, the team designed 5 different muscle architects: parallel, unipinnated, bipinnated, multipinnated, and convergent, each mimicking how real muscles are structured in the body. They used a technique called fresh 3D bioprinting to print detailed scaffolds made of collagen type 1, which is a major component of the muscle tissue. These scaffolds were seeded with C2C112 myoblasts, a type of muscle precursor. The printed architect helped guide these cells to align and fuse into the myotubules, just like the muscle. To see how well these engineering tissues were working, they looked at several different things: the myofiber alignment, the calcium imaging, force production, and finally in vitro vascularization to see how these tissue integrated and formed blood vessels after implementation. The results show that the myotubules aligned really well, with a printed scaffold geometry in the 5 architects. This means the design successfully guided how the muscle cells organize themselves. The tissues also respond to electrostimulation, which is a key function of a muscle. They showed calcium translate, which are changes in calcium level that trigger contraction, and they were able to produce both twitch and tectonic forces, just like a natural muscle fiber. The peak force was around 1 kilonewton per meter squared, which is within the expected range for engineering muscles, and showed good contractibility function. And importantly, when implemented in vivo, the host cell's blood vessels actually grew along the scaffold alignment, showing the structure not only guided the muscle cells, but also supported vascular integration, which is crucial for long-term survival and function. In summary, fresh bioprinting allowed precise control over the architect of engineering muscles, making it possible to recreate complex muscle structures like unipinnated and multipinnated patterns. The resulting tissues were both structurally and biomimic and functionally contractible, meaning they not only looked like the real muscle, but they also behaved like it, responding to stimulation and generating force. Importantly, these constructs also supported vascular ingrowth in vivo, which is essential for integration and long-term survival after implementation. Overall, the approach represented a promising platform for both regenerative medicine, like treating muscle loss, and for disease modeling in the lab, where we can study muscle function and drug response in a controlled 3D environment. Next, I'll be presenting on a foundational paper in skeletal tissue engineering by Dennis and colleagues, published in 2001. The study introduced the concept of myodes, or engineering 3D muscle tissues, and provided one of the first functional assessments of their excitability and contractility. It is considered a seminal work because its influence on both methodology and the direction of the field. So traditionally, muscle cells have been grown in 2D culture systems like Petri flat dish, but the problem is these systems don't replicate the real structure or behavior of a muscle tissue. Muscle is a 3D aligned and contractive tissue, something that flat cultures just can't recreate. Also, many previous engineering methods haven't included strong foundational benchworks like force production or electrical responses. So even if a tissue looks right, we don't know if it actually works like a real muscle. That's why we made a need for a 3D contractive muscle model, something that not only mimics muscle architect, but also performs like it. The kind of system that will be incredibly useful for both research and regenerative therapies. In this study, the team engineered myodes, which are 3D muscle bellies created in the lab. They were formed in either C2 C112 myoblasts, co-cultured with 10 T12 fibroblasts, or from primary muscle cells cultured from adult or neonatal rodents. Instead of using traditional scaffold, the cells were anchored between silk sutures and allowed to self-assemble and mature under tension. The tension is key. It mimics the mechanical environment of a real muscle and helps guide alignment and development. They evaluated several key features, including the cross-sectional area, the electrical excitability, twitch and tonic force, and relaxation time. Overall, this approach creates a simple but functional 3D muscle model without needing an external scaffold. All of the engineered myodes responded to electrostimulation, which confirmed that they were functionally contractible. When looking at specific force, the primary rodent myodes, those made from adult or neonatal muscle cells, performed best, producing 4 to 19 kilonewtons per square meter. In contrast, the C2, C112, T10 myodes consistently produce lower force, showing that while they can be formed muscle tissues, their functioning is not as strong. The C2, C10 based also had a weaker electrical excitability, along with a slower time to peak tension and a longer relaxation time, suggesting they're not as mature or responsive as the primary cell-based tissue. Overall, this study introduced the new term myodes to describe scaffold-free 3D muscle constructs that form natural between anchor points. It is one of the first to rigorously quantify functional properties like twitch and tonic force in engineering muscles, setting a new benchmark for future tissue engineering. Looking ahead, there are several key directions to developing electrical and mechanical stimulation protocols that can help further mature the tissue, making them stronger, more responsive. Integrating myodes into muscle-on-the-chip systems that will allow high thorough point testing and real-time analysis in more controlled environment. And finally, these models have strong potential for disease modeling, especially for muscle disorders, giving us a platform to study disease mechanisms and treatment in human tissue. So this slide highlights 4 internationally recognized leaders whose work has significantly shaped skeletal muscle regeneration and tissue engineering. Dr. Blau of Stanford is a pioneer in muscle cell biology. Her work focusing on rejuvenating aging or dysfunctional muscle cells to restore regenerative capacity, especially relevant in aging and muscle disease. Dr. Larsinen at University of Yukon is known for merging material science with biology. He was instrumental in developing fiber scaffolds for tissue repair and helped define the field of regenerative engineering, where biomaterials and stem cells converge. Dr. Cure from Australia investigated how much muscle stem cells behave genetically during development, repair, and disease. His team used zebrafish modeling to uncover evolutionarily conserved regenerative mechanisms. Insight from that informed human therapies. Dr. Glass, focusing on molecular pathways that regenerate muscle mass. He's also known for characterizing the myostatic pathway and targeting IGF signaling, laying the foundation for therapeutic approaches to counteractive muscle wasting.
All right, so actually, when you look at more locally, who's actually leading some of the areas of focus in this research, it's no surprise to see that the triangle has a heavy influence in this specific field. Starting off at Duke, led by Dr. Louis Edwin DeFronte, I believe that's how you say his name. They actually have a lab that focuses on in vivo biomechanics using an advanced radiographic and MRI technique. This enables soft investigation of soft tissue in both healthy and diseased states. Over at Wake Forest and their Institute for Regenerative Medicine, they actually have developed a computer-controlled system to engineer organized muscle implants in the lab, in an actual lab. This approach involves attaching muscle cells to collagen strands and subjecting them to cyclic stretching in a bioreactor, similar conditions that you would typically see in the human body. And even at our own faculty at BME, both NC State and UNC, there's faculty members investigating approaches to replace, engineer, or regenerate tissues and organs overall. Their work actually kind of encompasses more of a functional tissue engineering. Then they also use molecular biology, bioreactors, biomechanics, and a bunch of different techniques to kind of look at not just skin muscle tissue engineering, but also other organs as well. We're looking at the size and scope of the field. SMTE or skeletal muscle tissue engineering. It is a specialized segment within a broader tissue engineering field. It has experienced significant growth, but data when looking at the monetary value of this actual field, it's kind of limited. But you can actually get insights from the overall tissue engineering market. So after research looking at it, the global tissue engineering market was valued at 4.3 billion in 2022. It's expected to reach about 8.9 billion by 2028, which is typically an increase of 15.3% when looking at the compound annual growth rate. Investment and funding. So some of the biggest investments actually happen actually here locally. Starting at the bottom, Dr. Espen Spartanberger at ECU, actually down the road, recently received a $750,000 NIH grant to explore mechanisms that enable certain skeletal muscles to function without oxygen, aiming to hopefully develop therapies for ischemia-related conditions. The big one that, however, was up in Minnesota, the Metzger lab at the University of Minnesota. They received a $2.7 million NIH grant for their project titled "Skeletal Muscle Saccharin Function in Health and Disease." Now, when looking kind of at the more private sector companies and who's working on this specific field, you have both startups and established companies, which I think is nice because that's how you can kind of tell the growth of the field overall. Some of the startups include MyoWorks. They're creating engineered skeletal muscle tissues for implants. Tissue Dynamics. They developed a muscle on a chip and a neuromuscular model for pharma screening. And then some of the more bigger players in the field that have been around for a while include Organovo. They have to do stuff outside of skeletal muscle tissue engineering. I'm sure we've seen presentations in the past that have had this company as well. They use bioprinting, as Megan talked about earlier in the presentation. They use that for more pharma applications. And then Thermo Fisher, they supply muscle progenitor cells and differentiation systems for research and for industrial use. Some of the key images of SMT or skeletal muscle tissue engineering. First, starting on the top left, that's actually a 3D bioprinted it's 3D bioprinting of an actual skeletal muscle construct and has a sacrificial gelatin hydrogel component. Basically, this helps generate microchannels within the construct. These microchannels facilitate the diffusion of oxygen and nutrients to the cells of the construct in an effort to mimic these structures in vivo. And in the bottom right, I think this was the coolest part of my research. This is actually a recent publication in 2025. It's a robotic hand powered by a lab-grown muscle tissue that can perform, delicate hand gestures such as lifting a finger or slight rotations. It was a breakthrough from researchers at the University of Tokyo at the Waseda University in Japan. The hand itself is actually made. The hand itself is actually made of a printed plastic base, but the tendons have human muscle tissue, which help move the fingers as well. Looking at some of the key products under skeletal muscle tissue engineering, first one is the iPSC derived myoblast. Um, they're used in drug screening, toxicology, and disease modeling, and they're definitely sourced from different companies, but basically this helps to reduce the use of animal testing and improves the accuracy for human responses. Important one, hydro-based scaffolds. They're commonly used to culture muscle cells in a 3D environment. It offers biocompatibility and a moisture-rich environment for growth, just to try to mimic what an actual human muscle cell actually looks like. Fibrinogen and collagen are specific scaffold types that they actually use. And then how this all kind of ends is with muscle bioreactor systems. A lot of the industries use bioreactors to kind of simulate the mechanical forces, such as the stretching and the pulling, to kind of help develop and mimic kind of what muscle tissues undergo in our bodies every day. Looking at some of the intellectual property, counter ranges across the entire country. A team from Purdue, they have a patent for certain methods for creating 3D muscle tissue for restoration. Another team down in University of Central Florida, they have a patent out for nutrient mediums and methods to extend muscle cell culture viability. That's kind of the bit. I'll get into it later. That's kind of been the big limitation within this field is cell sourcing and cell viability to get it to last longer than what the industry standards are now. And then a team over at Rutgers has a patent in the area focusing on grafts. That helps promote integration between different tissues and it's essential for musculoskeletal function. That's also another one of the big limitations is that once they actually have found these cell sources, they've had trouble with having tissue crosstalk and getting these kind of tissues to kind of mimic exactly what our own muscles are like in our body. Some of the functional metrics, what these different companies and labs use to kind of test what their in vivo kind of muscle tissues look like. First off, it's not really a shocker, but contrail force generation, which in titanic forces, something I'm sure everybody in this class has learned through 301 and 302. Myofiber alignment and maturation, excitation, contraction, or EC coupling, fatigue resistance, and importantly, gene and protein expression as well. What is the definition of success in this field? Definitely some different mixed opinions. After doing a lot of research through different websites, different papers, this is kind of, I thought, what the best definition kind of for these areas were. So when looking at in vitro model systems, the definition of success would be really to get these myofibers maturation both aligned and functional and capable of force generation. For implantable therapies, vascularization is the name of the game. You want an inverted graft to integrate with host tissues and restore partial or full functionality. And then when looking at disease modeling and drug screening, you want faithful recapitulation of disease phenotypes and predictable response to different therapeutics, especially across different disease states. Now, what's kind of some of the current state of the art functional outcomes? First, when looking at contractile force generation, these engineered muscle tissues have achieved titanic forces ranging to 2.8 to 1.38 meganewtons. Definitely depends on different models and conditions. Force measurements have been reached up to 36.3 kPa. These, while are definitely improvements from what we've seen maybe 10, 15 years ago, these are still not quite to what the standard is for what we see in our human body today. Myofiber alignment, that's kind of this step one in all this whole area field. Engineered muscles exhibit aligned and mostly nucleated myofibers. Vascularization, as I mentioned earlier, they have to have the ability to integrate with host vasculature, leading to perfusion and survival of the tissue past just what they've gotten to be a week and a half so far. And of course, electrical stimulation affects our muscles obviously interact with our nervous system. So you need that chronic electrical stimulation of engineered muscle tissues to have increased myotubule size and enhanced force production and have some improvements on fatigue resistance as well. When looking at some of the applications for traditional skeletal muscle tissue engineering, first off, they look at volumetric muscle loss repair, basically used for traumatic injuries, trauma, congenital muscle loss, and typically these constructs are surgically implanted to regenerate muscle. This kind of goes into the second point of reconstructive surgery, massive limb loss, cleft palate surgery. These engineered muscles would help to restore soft tissue volume and function as well. Skeletal muscle patches for cardiac support. The heart's one of the biggest muscles in the body. Muscle grafts are used to support or mimic cardiac function post myocardial infarction. And then finally, in vitro muscle models for tissue testing, pharmacokinetics, toxicology. These models would help to evaluate the effects of different drugs on our skin and any potential side effects. Now, what I thought was really interesting was some of these novel, more radical skeletal muscle tissue engineering fields. Kind of the big ones that stuck out to me included actuators, meat alternatives, something a lot of us eat every day, textile and wearable biotech. There's actually a lot of research going on in different athletic companies trying to get wearable biotech to kind of increase the game for athletics. And then finally, injectable muscle progenitor therapies. They really don't have that currently. Nothing has been able to be specialized and injectable. It's always been, in an actual open surgery. And you can, with this slide, you can clearly see kind of the progression of this field when looking at the past and present methods. The biggest one, culture format, you're going from a 2D monolayer culture to now 3D aligned scaffolds. That's just kind of been the main thing with these past and present models. It's all come from 2D kind of environments. Now you're looking at 3D. We're looking at bioreactors. You're going from stipple static chambers to now you have more dynamic perfusion or stretch bioreactors to be able to really mimic what these muscles are going through in our body every day. And then really the last one I think, which is kind of important, it's been kind of a hot topic with ethics and biomedical and within biomedical engineering is the limited use of animals or rodents for these types of testing. Now you're doing these volumetric muscle loss models, in vivo, but not with the use of animals as well. Always important to kind of end this looking at the limitations because there is still limitations even today. In the past, you would have immature, non-functional tissue translation to animal model or human models, which is definitely kind of been the biggest bugaboo with all these kind of engineering fields. I feel like is just being able to fully replicate what the human body looks like. So now, presently, you still have some of these issues, but it's still kind of a little bit lagging behind. But I think the next step for this field would be to get more adult muscle cells to both, mimic both the endurance kind of factor of muscles, force generation, and be able to respond to electrical stimulation as well.
Okay, so in skeletal muscle tissue engineering, traditional methods usually involve seeding the myoblasts or satellite cells into biodegradable scaffolds. But several research groups are exploring more non-traditional strategies, which are approaches that break away from this scaffold cell paradigm and often borrow from other disciplines and rethink these problems from a new angle. So first, we're going to look at what's been mentioned a couple times, but biohybrid muscle systems or a muscle on a chip, which is one emerging strategy that involves creating biohybrid actuators. And these are micro-engineered platforms where muscle cells are integrated into flexible synthetic substrates to form small contractile units that are mimicking this type of muscle movement. And the goal isn't always therapeutic regeneration, as it's also to model function or serve as soft robotics, such as the image that you see there on the right. And these systems respond to electrical stimulation and generate force, which makes them useful for drug testing or even powering micromechanics. And the scientific basis here is rooted in mechanoelectric feedback and biorobotics, which are treating muscle not just as a tissue to regenerate, but as a bioactuator that can be engineered in modular form. Moving on to another non-traditional strategy being scaffold-free muscle constructs. In these approaches, muscle progenitor cells are cultured in such a way that they form spheroids or aligned bundles without the use of any synthetic biomaterial. These constructs often rely on extracellular matrix deposition from the cells themselves to stabilize the structure. The benefit here is that they most closely replicate native tissue architecture and avoid foreign body responses that can occur with scaffold materials. The rationale is biological, as native muscle tissue is incredibly organized and dense, and introducing a synthetic scaffold sometimes disrupts that organization. By letting cells self-organize, researchers aim to better mimic how muscle forms during development. And a third non-traditional method is 3D bioprinting with neural integration. So traditional skeletal muscle tissue engineering methods focus mostly on muscles, but more recent approaches are using co-printing techniques that include the motor neurons and vascular cells in the same construct, which aren't usually included. And this shift toward multi-tissue printing acknowledges that skeletal muscle function depends not just on the muscle fibers, but also on the innervation and blood supply it has. Some studies now include neural spheroids or even induced pluripotent stem cell-derived neurons to try to recreate a neuromuscular junction as seen in the body. The scientific justification here comes from developmental biology. Muscle fibers won't fully mature or contract effectively without proper electrical and chemical communication from nerves. So instead of engineering muscle in isolation, these approaches look at the muscle as part of a broader functional system. In skeletal muscle tissue engineering, bioreactors play a crucial role in mimicking the dynamic physiological environment that muscle cells need to differentiate, align, and mature. Whether in vitro or in vivo, these systems are designed to replicate key stimuli like electrical pulses, mechanical tension, and nutrient flow that are all essential for building functional contractile muscle tissue. So we're going to walk through three main categories. The first being in vitro bioreactor systems, which are widely used to promote alignment, contractility, and myogenic differentiation, as I mentioned earlier. Skeletal muscle cells are particularly sensitive to physical cues, and these systems aim to recreate that type of environment. Some bioreactors apply cyclical mechanical strain, as you see the fourth category there on the image on the right, which is stretching and relaxing the engineered muscle tissue to simulate normal loading. Others use electrical stimulation, such as that third category, which helps cells form sarcomeres, increase contractile strength, and even respond to action potentials. And a great example of these is a dual stimulation bioreactor that applies both mechanical stretch and electrical pulses, which has been shown to significantly enhance that myotube formation and force generation. And these setups help muscles contract mature beyond what's possible in static culture dishes. Moving on to in vivo bioreactor concepts, these use the body itself as the environment to mature engineered muscle constructs. For example, researchers might implant a scaffold seated with myogenic cells into a well-vascularized area, like under the skin, to allow host cells and vasculature to infiltrate that construct before it's transplanted to the injury site. Some advanced strategies use arteriovenous loops to pre-vascularize the construct in vivo, giving the muscle a functional blood supply before it's moved to repair a volumetric muscle loss defect. These in vivo systems are especially valuable because they address one of the biggest challenges in skeletal muscle tissue engineering, the need for vascularization and integration with host tissue. They also expose the construct to the immune system and mechanical forces in a way that's difficult to replicate in vitro. And for our third, which we have mentioned multiple times on a broader scale, and this will dive a little into some specifics, but that being the muscle on a chip system. And these offer a way to simulate muscle behavior in a highly controlled and miniaturized environment. These platforms often feature one, aligned muscle bundles seated in microchannels, two, integrated electrodes for stimulation, and three, force transducers, force transducers to measure contraction strength. They're especially useful for drug testing, such as modeling muscular diseases, or studying how muscle responds to stress, fatigue, or even repair signals over time. Some chips even integrate motor neurons or vascular channels, allowing for the recreation of neuromuscular junctions and perfused environments, making these tools valuable not just for basic research, but also for preclinical testing of these therapies. So overall, bioreactors are a lot more than just growth chambers. They're this type of functional bridge between biology and biomechanics, helping us further our research and understanding of muscle tissues. Moving on to the artificial intelligence section of the presentation, one of the most valuable skills we can have is asking intelligent questions of AI systems, and with that, critically evaluating the answers we get back. Um, these are growing tools within the field, and it's extremely important for us to understand how we want to use them. So for this slide, I used Perplexity AI, which is one of the better performing platforms in terms of non-hallucinatory responses and these linked references to the literature. And I asked it two questions as we were instructed. The first being, what are the most important advances in the field in the past 5 years? And these are three responses that you see here on the right, being bioengineered constructs with integrated vasculature, 3D bioprinting techniques, and scaffold-free self-assembling muscle tissues. And I found these to be the most critical and repeated answer among the chat system. And there were a few other mentions of things like human-induced pluripotent stem cells and mechanical and electrical stimulation protocols that were less frequently cited but still discussed. And most of these sources were from Nature Biomedical Engineering, Biofabrication, and recent reviews in Acta Biomaterialia. Moving on to the second question was asked, when will functional engineered tissues be commercially available within this field? And these are the two main responses that I wanted to highlight, which are small scale engineered muscle grafts would be expected within 5 to 10 years. However, large load bearing muscle constructs would be more foreseeable in 10 to 20 years. And not mentioned here on the slide that I just want to discuss was that they were saying that the pace of this translation depends heavily on regulatory approval and clinical trial data. And they also mentioned the lack of FDA-approved full-scale skeletal muscle implants in general in the field today that could limit some of this research. So in terms of analyzing the responses that we got from this AI chat, there were about four things I wanted to highlight. So I would say in general, this was a surface level synthesis, not in a bad way, in a way that that's how we asked it. We asked it a very general question, and the answers provide good context, but not specific detail. For example, the part where it discusses integration of vasculature and nerves is mentioned as a success, but it doesn't exactly explain how it's being achieved or what the failure rates are in these animal models it's highlighting. Second, I would say there's somewhat optimism in these future deadlines or approaches that we're hinting at. Saying that we'll see functional graphs in 5 to 10 years may be technically true in niche applications, but in general, I would say it overlooks the scaling and manufacturing barriers that are still unresolved for these larger muscle defects. Third, a lack of discussion on muscle functionality. The AI didn't discuss the actual force generation or fatigue resistance or contractile behavior of the engineered muscles, which are all essential to whether something is considered clinically useful or not. And lastly, there was no mention of failure or roadblocks, such as cell sourcing and construct remodeling over time, which complicate the field heavily and all of which slow clinical translation and would have been helpful in understanding this timeline more specifically. Skeletal muscle tissue engineering in general has made impressive progress in the last 2 decades, but we want to look at the reality of the situation. So we'll start with the past, which early work in this field focused on relatively basic constructs, such as seeding myoblasts or satellite cells onto biodegradable scaffolds, with hope for fusion into these myotubes. These studies provide proof of concept that muscle cells could survive, align, and even generate small amounts of contractile force in vitro, which is great. These in vivo models emerged shortly after, but the results were limited by issues like poor vascularization and lack of innervation after implantation. Most constructs failed to regenerate large volumes of muscle tissue or restore meaningful function in these injury models. Moving to the present day, we've moved well beyond those early approaches, and researchers are now integrating electrical stimulation, mechanical loading, and bioreactors to promote this muscle maturation. Some labs are developing pre-vascularized constructs, while others are incorporating motor neurons to recreate these neuromuscular junctions. We're also seeing the use of induced pluripotent stem cells for patient-specific muscle tissues, and 3D bioprinting is allowing for precise alignment of fibers. There are now successful preclinical studies showing improved force recovery in animal models of volumetric muscle loss, especially when using multicellular innervated constructs. However, clinical translation remains very limited. There are still no fully functional, off-the-shelf engineered muscle implants approved for human use. Looking ahead, the future of the field seems headed in the right direction, um, and we're trending more towards biologically integrated systems that don't just mimic muscle and innervation, but attempt to restore function through vascular integration, force transmission, and innervation. Um, so the larger questions at the end of the day, is the field really progressing? Yes, but at somewhat of a slow climb. The science is becoming more refined, but there's still a large gap between these lab-grown muscle bundles and clinically viable therapies that we're aiming towards. So, what are a couple things that could improve these future prospects? One, stronger collaboration with clinicians, ensuring that these constructs are surgically relevant and tailored well. Two, focus on modular systems. Instead of chasing these integrated constructs from the start, we might gain more ground by assembling systems piece by piece, such as muscle plus nerve plus vessel, and merging them functionally. Third, investment in large animal studies. This would help smooth out the path towards translating these to human trials. And fourth, realistic goals, setting these realistic stepping stones rather than aiming straight for full replacement of load-bearing muscles could help iteratively progress the field forward. Moving on to the play of hype in the field, as much as we just talked about this realistic progress, how much of it is hype? So, when looking at this said hype, we want to first look at the facts. And yes, there has been real measurable advancements in this field, such as the ones I mentioned prior, engineered constructs, neuromuscular junction formation, and achieving functional recovery in animal models. And these are important milestones to consider. But at the same time, the way some of this work is communicated to the public is either exaggerated or somewhat misinformed. In many cases, the engineered muscle being reported on is only a few millimeters long, um, and has been tested in something like a rodent and may only generate a fraction of the force of what a native muscle needs. But claims like ready for transplant or lab grown replacement muscle are often thrown around the field. And these statements can paint a picture that we're just months away from off the shelf muscle implants. But in reality, we're still struggling with things like I mentioned reliable vascular integration, consistent innervation, and long term viability. Um, so is the field dominated by hype? I would say not entirely. Most researchers are still doing careful work, trying to get at the core science and moving this field forward. But the narrative around the field, such as those with scientific publications, and news reporters that move outside of these scientific publications can definitely skew this field towards the idea of hype. So, what can we do to fix this? We can be a lot more transparent, such as the novelty, publishing null results, even if there are no results, still important to let people know maybe that that's not a path that we should go down forward. Being clear about what stage the research is actually in, being honest that we're not weeks away from this new finding that we aren't, being transparent about that, and designing public messaging that balances this potential with patience. Um, with this idea of progress and hype, we still want to look at the barriers that are face or stopping the field from progressing in general, or at least slowing it down. And it broke it down into a few different ones. First being technological barriers, and the first barrier within that is scalability. And we can now create small engineered muscle tissues that look promising in vitro, but scaling those constructs up to clinically relevant sizes is just extremely difficult. Muscle is structured, anisotropic, and load-bearing, and with all of it, it's a complex tissue. We need constructs that not only match the size and shape, but also mimic its mechanical properties, transmit force effectively, and survive under stress. And then there's vascularization and innervation, and without a sufficient blood supply, implant and muscle tissues undergo necrosis. And with that, we need solutions. Um, in terms of scientific barriers, biologically, we still have a lot of unanswered questions. We don't fully understand how to recreate the complexity of native muscle development, especially when it comes to neuromuscular junction formation. There's also a lack of long-term data. Most studies focus on short-term function, such as days to weeks, but we still don't understand how these constructs behave after months or years, even in vivo. Will they integrate? Will they remodel? Will they trigger fibrosis? We're not fully sure. Additionally, there's problems with cell sourcing. Um, allogenic cells raise immune compatibility issues, and immune pluripotent stem cells hold promise, but the differentiation protocols are variable and still maturing. And lastly, there's some of these conceptual barriers that are more philosophical. And there's a tendency to equate form with function and to assume that if a tissue looks like muscle, it'll act like muscle. But functional outcomes like force production are much harder to engineer than structure alone. There's a bias towards complexity for complexity's sake, simply saying that by recreating biological complexity, we're doing the job better, which is not true. A construct that works beautifully in the lab may be impossible to implant, too expensive to scale, or even incompatible with surgeries. And if it can't make it to the patient, it's not really a solution. So we would like to spend a lot more time into these errors of thought to remove the barriers and lead the field forward. Now that we looked at the barriers, oops, I've lost the slideshow. I'm sorry. One second. I can still share. I think the slide is back up. Sorry. I will. My fault. We are back. So in terms of these opportunities, I just laid a few out as to how the field can move past these boundaries and move forward. So first, focusing on smaller and high impact clinical targets. Instead of trying to replace entire muscle groups in the limbs, we can target smaller scale but higher impact areas such as facial muscle repair or urethral muscle reconstruction, or even vocal cord and diaphragm repair. These targets require less bulk tissue and lower force output and are often more forgiving in terms of integration, what we would hope. Two, improving pre-vascularization and innervation strategies. We can make real progress by refining modular systems that separately develop muscle and neural components. And these strategies are already being tested in animal models and could be optimized for translation. Third, establishing functional standards for engineered muscle. One of the most practical things we can do right now is set clear performance benchmarks, such as what percentage of native muscle should be restored, how much fatigue is acceptable, and things along those lines. And creating these standardized metrics would help compare results across studies and accelerate regulatory approval down the line. Four, expanding use of advanced in vitro platforms. We've already mentioned these muscle on a chip systems, and there's some underused opportunity. They have a lot of benefits, and with the addition of force sensors and electrical inputs, these systems can simulate fatigue, training, or disease states, and offer a lot of valuable at extremely low cost. And lastly, even with the use of generating some AI prompts within these presentation instructions, being able to leverage AI and machine learning for design optimization is a large opportunity for this field. AI could help optimize scaffold architecture, stimulation protocols, and even predict construct behavior over time. Uh, these tools won't replace the biology, but they'll help us design smarter and faster with fewer dead ends. Which is much needed in the field. And lastly, we want to take a overview of the milestones that this field has experienced from the past to the present, and even what we continue to do to try to just give a general overview of where we're at and some important things we would like you to take home with this presentation. So first, in 1994, we begin with the first index publication in skeletal muscle tissue engineering. This marked the start of a very slow phase. During the 1990s and early 2000s, most studies focused on basic myogenic cells like satellite cells and myoblasts, and early scaffold designs using natural materials like collagen. And the goal was pretty simple here: create aligned myotubes that could survive in vitro. Moving on to 2005 to 2010, researchers started introducing mechanical and electrical stimulation into engineered muscle systems. And this was a pretty major turning point for the field, as applying stretch and electrical pulses led to much better alignment, sarcomere development, and functional maturation of these engineered myofibers. Moving on to the next 5 years, during this phase, the field expanded into new cell sources, such as bone marrow-derived mesenchymal stem cells, and began experimenting with composite scaffolds and biodegradable polymers. This few years saw the early emergence of scaffold-free approaches where myogenic cells could self-organize, as I mentioned earlier. And 2015 to 2020, the field accelerated heavily, and the timelines that I were looking at were describing this as the beginning of the biofabrication era, and publication counts rose dramatically, and 3D bioprinting entered the spotlight as researchers developed increased complex constructs with spatial patterning, hybrid materials, and even vascular and neural components. Things like electrospinning, hydrogels, and bioprinted muscle sheets became pretty common in the field. And moving on to the where the field's been for the past few years, and even today, um, we've moved beyond structure, mimicking structural components of the muscle into building muscle that's vascularized, innervated, and capable of force generation. Some groups are even using these um, pluripotent stem cell derived muscle cells for personalized therapy, and the development of muscle on a chip platforms is allowing us to test drugs and model diseases. Um, and moving on to milestones that are not yet achieved that we do hope to be tackled in the near or later future. that could help steer the field in a positive way. One being stable neuromuscular junction formation. We can form these neuromuscular junctions in vitro, but replicating these connections in vivo and keeping them functional in the long term is still a major hurdle that we would like to tackle. We've also yet to engineer vascularized muscle grafts that maintain long-term perfusion, especially when scaled up to these clinically relevant sizes. Um, and lastly, from a clinical perspective, there's still no FDA approved off-the-shelf skeletal muscle products for implantation. Everything remains in early stages or investigational use. So overall, this timeline reflects a field that's gone from early, more simple techniques to these extremely developed and highly, sorry, techniques with high potential, clinically speaking, to transform the field. And with that, we have all of our citations listed here and appreciate you guys for listening to the presentation. Thank you all.